Thank you so much, Jeremy, for such a kind introduction. I feel like I don't even have to give this talk anymore because you gave a nice overarching view of some of the things I'll be speaking about today. And I'm really very honored to be asked to be a keynote speaker at this Congress. So thanks very much to the Congress organizers for inviting me. What I would like to do today is give you a very broad overarching view of much of the research that my laboratory has been working on in the past 19 or 20 years at the Denver Museum on camel spiders. And as this audience knows, if I can forward my slide. As this audience knows, camel spiders are arachnids in the order Solifugae, and they're well known for having very large, powerful jaws. They will run down any prey that they think they can get, and they will tear that prey apart with those enormous jaws. They have incredibly high metabolic rate, much higher metabolic rate than probably any other arachnid group. And they're known, unfortunately, by more common names than I'm sure any other arachnid, including camel spiders, wind scorpions, children of the earth. In parts of Africa, they're known as haarskieters, which means hair cutters, because it's believed they come into your bed at night to cut your hair with their very large jaws. Yagan spinacopa means chasing spiders because of their very high metabolic rate and fast running speed. In some parts of Mexico, they're known as Madre de Alacran. In other parts, Perito de Agua. And I'm very embarrassed to admit that I added another common name, which is Spawn of Satan, because they are a very challenging group to work with. They're difficult to collect, hard to find in the field, very, very challenging to keep alive for any length of time in the laboratory, and nearly impossible to raise from hatch through maturity in the laboratory. So it's very difficult to get a laboratory colony established. Nevertheless, I'm very pleased to say that a lot of different labs are publishing more and more on the biology of this group of animals. So there seems to be a lot more interest and a lot more success in doing research on these animals. We've even seen at the Congress, David Retch's talk on sexual selection in ammotrachids, and I know Alfredo Peretti and Eileen Hebetz are working on the group, and there's a poster presentation as well on ammotrachid sexual selection. So there's a lot of papers that are coming out. A lot of these are on the morphology or the systematic, not so much on the systematics, but the morphology and the taxonomy of the groups. So this is great to see. I think we're seeing this field of study moving forward. So to give you an idea, just what we do know about camel spider development, we'll start there since birth is a good place to start with any animal. So what we do know about them, we don't know very much about many, many aspects of their natural history. So many aspects remain a mystery to us. However, what we do know is that the females will lay their eggs, most likely in burrows. And you can see this female upside down with a cluster of eggs. They hatch out of those eggs as very helpless, very pale offspring. So the first embryos are just clustered. They don't move very much. And it's not until the second instar nymphal stage that we begin to see sclerotization, hardening of the exoskeleton. And by that second instar stage, they also begin to feed on their own and take down very small arthropod prey like Columbella, proturans. And at that point, this is one of the reasons they are challenging, is if in the laboratory you can get them, first of all, if you can get the female to lay eggs successfully, that's a miracle. And if she does lay eggs, usually they are inviable. If they are viable, oftentimes they will die before they hatch. If they do hatch, it's very difficult to get them past the first instar stage. If you get them to the second instar stage, you have less than a day to separate them all out. Otherwise, you just end up with one very fat sulfugid. So the cannibalism rate is very high at that point. Solifugae or camel spiders are found in desert ecosystems throughout the world, except Australia, New Zealand, Madagascar, but they're found commonly throughout desert environments in the world. Solifugae. There are two Nearctic families, the Arachnidae and the Eremobatidae. And my lab has been focused on the biology of the Eremobatidae. And this is a slightly old slide. At this point, there are about 190 or so species of Eremobatids. So we have described several new species since I created this slide. In 2018, myself and my colleague, Matt Graham, shown behind me, we received, he's co-PI on a big National Science Foundation grant that we received to explore and revise the systematics, taxonomy, look at the biogeography of Eremobatidae. And one of the reasons we got this grant is because I am fortunate enough to work closely with a research associate named Jack Brookhart. And Jack Brookhart, you may know his name because he received his Master of Science degree in the 1960s from Martin Muma. So Jack is truly the world expert on this order of arachnids. Jack has been publishing on this group constantly since the 1960s. Through this grant, we also were able to recruit two graduate students, my doctoral student, Erica Garcia, and Ryan Jones, I recruited as a master's student. I'm very pleased to say that Ryan just recently finished his master's degree looking at the phylogenetic significance of one morphological character in Solifugae. And I'll touch on both some of the morphological work Erica and Ryan have been working on later in today's talk. Ryan has decided to stay on and work on his PhD in my lab. We also are working with Warren Savary, who is updating our online site, our online information site and website about Solifugae. And through the grant, we were also fortunate to recruit a postdoctoral scholar, Carlos Santabañas Lopez. And Carlos, I'm happy to say, is now an early career professor at a university in Connecticut. So Carlos and Matt presented their poster at this Congress, and I'll talk about their work again later in this talk. We're also able to recruit through the grant several undergraduates. This is one of the undergraduates, Lance Herrera, and we recruited Felix Chaniago, and my REU students, these undergraduates, have been helping in the field, but also in the laboratory, and I'll talk what they're doing for me. And I'm collaborating with Edmundo Gonzalez Santillan, and he has also recruited some undergraduate students who are working on this group. So we're getting a nice big team of people together to look at this family and this group of arachnids. So this slide presents some of the overarching goals of our National Science Foundation project. So as Jeremy mentioned early on, in 2015, we published the first molecular phylogenetic analysis of the family of Eremobatidae, and I'll show you those early results in a moment. But what we want to do now is that was sort of using your classical multi-locus approach, and I was able to use a target taxa approach that represented all the different genera within the family. But what we want to do now is use next generation sequencing and UCEs which will enable us. If you saw Hannah's talk, she mentioned that one of the great things about UCEs is one of the first step is chopping up your DNA. So it's really great for us museum professionals because UCEs allow us to increase taxon sampling by using the taxa that are in museum collections. So we should be able to greatly increase our taxon coverage. And hopefully this new phylogenetic analysis will resolve some of the unclear clades that were expressed in the 2015 approach. We plan to then use that new phylogeny to totally revise the taxonomy and do a taxonomic revision of the entire family. And we're doing that in sort of a piecemeal approach. So Erica, our PhD student, is responsible for doing some of the clades some of the groups of Eremobatids. Ryan is responsible for revising the phylogeny and the systematics and the taxonomy of another couple of clades. And through all of this process, we have also been exploring the phylogenetic and functional significance of various aspects of their morphology. Sulfugids have a lot of really interesting morphological characters, and I'll touch on all of these different approaches in the course of today's talk. And we're also serendipitous about our research. So if we observe there's so little known about the natural history of camel spiders that we take advantage of exploring questions as they arise. So we also are documenting behaviors that are unique to sulfugids, trying to better understand their ecology, et cetera. And then Matt and Carlos have been responsible for carrying out the first robust phylogeographic study of camel spiders. And that work is being presented at this Congress this year. So hopefully he presented the poster, Carlos did, on Monday, but it's also available for viewing. If you didn't catch it, please do. So it's poster SB020, and it's looking at using RADseq to reveal patterns of diversification and introgression in one genus of North American camel spiders, the Eremocasta, and using that to better understand the evolution of desert ecosystems. So this gives you an idea of some of the type localities through North America where we find different species of Eremobatids. And Eremobatids are actually found, they are distributed from southern Canada all the way through southern Mexico. So in the past several years, our lab has been visiting sites in Western North America. Last in 2019, Matt and Carlos and their crew visited Baja Peninsula. We visited and collected in northern Mexico to collect fresh material and also to document new species. In our trip to northern Mexico, we collected what will represent probably about 10 to 12 new species in the family of Eremobatidae. So I said in the beginning that this is a challenging group to collect in the field, but it is getting less and less challenging after a couple of papers that we produced in 2018 and 2019. So with Edmundo Gonzalez Santillan, we published a paper in 2018 that demonstrated that you could use illuminated pitfall trapping, and that was a really effective method for collecting sulfugids in the field. And it has been known for decades that Solifugae seem to be positively phototactic. So we developed this very simple kind of pitfall, illuminated pitfall design where you use landscape fencing that will funnel the sulfugids into pitfall traps that are scattered around this array, this pitfall array, and then you hang lights over your array. The lights actually attract the sulfugids to the array. Now, are they attracted to the lights, or are they attracted to the signals that are being produced by the insects that are attracted to the lights? The vibrational signals and the pheromones, we don't know. But in a paper that Matt and his crew published in 2019, he did a really nice little comparative study where he set up illuminated and non-illuminated arrays at several different sites in the North American desert. The only difference between these arrays is one was illuminated, one was not. And he achieved incontrovertible data that indicated that the sulfugids were only coming to the illuminated arrays. So it is now getting a bit easier to find and collect these animals in the field. We use these pitfall traps that are basically just Tupperware containers that have propylene glycol in them to collect the sulfugids. We leave the traps out overnight, pull them out of the Tupperware containers and put them directly in 95% ethanol. So this is just some field shots showing our different crews working in Mexico and in the Baja Peninsula and in northern Mexico. So to understand the work that we've been doing, you have to understand a little bit about the taxonomy. So the family of Eremobatidae, prior to our 2015 analysis, was subdivided into two subfamilies, the subfamily Eremobatidae and the subfamily Therobatidae. There were 5 genera placed within the subfamily Eremobatidae, the genera Eremobatides, Eremocosta, Eremorax, and Eremothera, and Horobatides. And within the subfamily Therobatidae, there were 3 genera placed there, Chambria, Eremochelys, and Hemerotreca. And the diagnostic character that was used to differentiate these 2 subfamilies was the number of tarsal claws on their very delicate front legs, first legs. So in the Eremobatidae, in the subfamily Eremobatidae, they were known to have, they were known to have 1 tarsal claw on the first leg, whereas in the Therobatidae, they were known to have 2 tarsal claws. So one of the things I did early on, and I should say also, please note that the 3 very large species-rich genera, Eremobatides, Eremochelys, and Hemerotreca, were themselves subdivided into various species groups. So you have to understand a little bit about this taxonomic designation. So one of the things I did early on in the grant work, the recent grant work that we got, was I used SEM, which had never been used before, to really establish that this claw pattern held true for these different genera. And so I just used SEM to look at the number of claws on the first leg within these different genera. And what I found is that in the 3 genera that supposedly had 2 claws on the first leg, only 2 of those genera truly do have 2 claws, Eremochelys and Hemerotreca. When you really look closely at Chambria, it has only a single modified claw. When you look at the number of claws on the genera that should have one modified claw, you find that for the most part, those genera do in fact have only 1 claw, except for Horobatides, which is only has maybe 2 species in that genus. And Horobatides, it has a very modified split claw. It doesn't have 1, doesn't have 2, but the tip of that claw is split into 2. So the claw characteristic using SEM is not a good character. Is not a good character. And the phylogenetic analysis we published in 2015 clearly demonstrated that that 2-clawed subfamily is artificial. And I'll show you that in just a second. But in this 2015 phylogenetic analysis, we used your traditional multi-locus approach. We used 2 different mitochondrial loci, 2 different nuclear loci. We used 81 target taxa, and those target species represented all 8 genera within the family Eremobatidae. They included 14 of the 18 species groups within those 3 really big genera, Eremobatides, Eremochelys, and Hemerotreca. We used 3 different families as outgroups, Cartiidae, Gylipidae, and Amortrecidae, as a way to test the monophyly of the family Eremobatidae. And Matt was also involved in this project, and he used beast analysis to propose a time-calibrated phylogeny for the family. So one of the things this earlier phylogenetic analysis showed is that the family Eremobatidae does seem to be a well-supported monophyletic family. So that's good. But when you look at that 2-clawed subfamily that included Eremochelys, Hemerotreca, and Chambria, it turns out that those genera are paraphyletic and polyphyletic. They're coming out all over the tree. And I know from the SEM work that claw characteristic is a poor characteristic to use for the monophyly or for the designation of those genera. Whereas the 1-clawed genera, except for a couple of situations, most of the 1-clawed genera are coming out monophyletic, except for Horobatides, which from the SEM work, it was clear has a split claw, and except for Chambria, which we thought had 2 claws, but it turns out has only 1. So again, that claw characteristic is not a good diagnostic characteristic. When testing the monophyly of the different genera, some of the genera are coming out monophyletic, like Eremarax is monophyletic, Eremocasta here is monophyletic, Eremathera is coming out monophyletic. Chambria, in some of our analyses published in 2015, came out as a strong, not shown here, but it's a strongly supported genus. But the species-rich genera, Eremobatidae, Eremachilus, and Hemerotreca, are paraphyletic or polyphyletic. So that is what we want to do now, is we want to use this as our starting point for increasing taxon sampling using UCEs, which will increase our data set to try to resolve some of the messiness that we see in this early phylogenetic analysis. The species groups, only a few of those 14 species groups that we tested are coming out well supported. Eremobatidae scaber is a well supported group. Eremobatidae palpes is well supported. Part of the Eremobatidae palpicigellosus clade is well supported, and Hemerotreca banksii is well supported, but the other species groups are not well supported. My master student, who's now one of my PhD students, Ryan Jones, is going to revise the systematics and phylogenetics and taxonomy of these groups, of this clade, whereas Erica is going to try to tackle all the genera that had been placed in the Therabatinae. With Matt's analysis, his beast analysis, what he found in this time-calibrated phylogeny is that it suggests that Eremobatids originated in the Cenozoic, with early diversification probably occurring in the Oligocene as steppe and semi-desert habitats begin to replace forests and savanna habitats in Western North America. So that was a very nice result, because it suggests that as desert habitats appear, we begin to see the occurrence and divergence of this group of desert-adapted arachnids. And we hypothesize that early Eremobatid diversification is associated with the complex geologic history of Western North America and the resultant changes in aridity. And that's the work that Matt and Carlos are using RADSeq to explore, is that hypothesis of using one genus, Eremocasta, which is well-supported genus, and seeing if you can understand the diversification of the species within that genus as it maps onto the diversification of desert habitats. So do check out his poster. This is a little snippet from his poster. So the conclusions of that earlier study are that the subfamily Eremobatidae, which is defined by having a modified single claw, is relatively well supported, except for the split claw Horibades and the one-clawed Chambria. Whereas the subfamily Therabatinae is polyphyletic and is not well supported. Some of the genera, Eremocasta, Eremarax, Eremathera, Chambria, are well supported. The other genera that have more species in them, Eremobatidae, Hemerotreca, and Eremachilus, are polyphyletic or paraphyletic. Some of the species groups are well supported. The others, most of the others, are not. And the beast analysis provided some jumping off information that we can use to dive into and explore in much more detail the diversification of Eremobatids and how that diversification relates to the evolution of desert habitats. So what I want to do for the rest of the talk is speak about some of the work that we've been doing and that we've been doing for the past 20 years on the morphological characters of Solifugae. And a lot of these characters are involved in are related to the chelicerae and to the pedipalps. But Solifugae have a lot of really unique and interesting morphological characters, including malleoli on the underside of the fourth leg. And these malleoli are analogous in function to the pectines of scorpions. So they drag those malleoli on the ground, and they're most likely picking up, there's evidence that they're picking up chemical cues, probably from prey, from the ground. They have sexually dimorphic chelicerae, and I'll talk a lot more about those sexually dimorphic chelicerae in a moment. Ryan Jones has looked at the phylogenetic significance of these spiny setae that are found on the underside of the abdomen of males, another sexually dimorphic character, and these structures are called conidia. Erica has been looking at the phylogenetic significance of palpal spines that have been used as diagnostic characters. And we've explored some of the setae that are found on the pedipalps of Solifugae, because there's a lot of information there, a lot of phylogenetic information and taxonomic information in those hairs and those setae that are unexplored. So one of the earlier studies I did, as Jeremy mentioned in his very kind introduction, was looking at the function of the suctoral organs, which are these balloon-like structures that Solifugae can use to climb up or grab hold of smooth surfaces. And those suctoral organs can be exuded from the tip of the pedipalp and pulled back in. So we did some light microscopy and SEM work to better understand the function and how those suctoral organs work. And what we found is that the surface of the suctoral organs act like the pads of geckos. They form a hydrophilic bond to smooth surfaces. And that's the organ that solifuges, if you've ever tried to keep a solifuge in a glass jar or a glass terrarium, they most likely climb right out, and they're climbing out of that smooth glass jar using those suctoral organs. So their suctoral organs form a hydrophilic bond and allow the animal to get to climb right up those surfaces. Rodrigo Willimantic did a really nice study showing that they also use the suctoral organs to hold on to prey and pull the prey closer to the chelicerae. So what we found is that the suctoral organs are pushed out of the tip of the pedipalp using an increase in hemolymph pressure, and they're pulled back in using tendons and musculature that pulls it back into the pedipalp. And Anya Klon also followed up our study with a much nicer study using much more classy histological techniques to look at the suctoral organ. Klon's paper came out in 2008. We've also surveyed with a former undergraduate who's now a doctoral student, Patrick Casto. We surveyed the setae that are found on the pedipalps of all the different families of Solifugae, just to get an idea of the diversity that is present in those setal structures. And then we focused in on one sexually dimorphic setal structure called papillae. So the papillae are a field of little Christmas tree-like structures, specialized setae, that are found on the underside of the male pedipalp, sexually dimorphic, of three different families, of the family Eremobatidae, Cartiidae, and Solifugae. So we wanted to better understand what the functional significance of these specialized setae might be. So we used SEM, TEM, and we worked with Doug Gaffin to also use electrophysiology to explore the functional significance of these structures, and also to understand if there was a phylogenetic significance to them. And what we found using SEM is that all the species within the family of Eremobatidae that have papillae on the pedipalps of males, all those papillae, no matter what species you look at, look like this. They look like miniature, very dense Christmas trees. Whereas all the species of Cartiidae have papillae that look like this, and all the species of Solifugae that have papillae, all their papillae look like this. So there is a phylogenetic significance, but only at the family level, not lower than that. So we tested functional hypotheses. We wanted to see if these structures might serve as, since they are sexually dimorphic, we hypothesized that they may serve as chemoreceptors or mechanoreceptors or olfactory receptors. So we use TEM and electrophysiology to test these different hypotheses. This is just showing a cross section through a papillae, showing these spicule branches. And using TEM, we were able to demonstrate that there's a dendrite that runs through the shaft of the papillae. And that is what you would expect for a chemoreceptor or olfactory receptor. I don't have a slide of it, but we also saw evidence of pores, openings to the outside, which is again what you would expect for a chemo or olfactory receptor. We found tubular bodies at the base of the cedal shaft, and tubular bodies are what you would expect of mechanoreceptors. We then used electrophysiology in Doug Gaffin's lab, and we used electrodes on a live male, poor live male Solifugae. We stuck the electrodes at the base of the pedipalp, and then we exposed that pedipalp and the papillae on it to various chemical cues. We pushed the papillae with a little capillary to see if it resulted in a mechanoreceptor signal. And we also, so we tested it with chemical cues and tested to see if it was a mechanoreceptor. We also exposed it to heat and water vapor. And what we found is that with the TEM work and some of the electrophysiology work, demonstrated that there is evidence supporting the hypothesis that these structures are chemoreceptors and mechanoreceptors. So pushing it did result in an electrophysiological signal. But there was no support for them being thermoreceptors or hygroreceptors, and no support, no evidence of a gland at the base of it. So no evidence that they are secretory organs. And this work was published back in 2014. Erica has been looking at the spines on the pedipalps of Solifugae. And one of the reasons she's interested in the spines is because, in particular for the genus Eremobatidae, one of the reasons 2 or 3 species are placed in this genus is because the males and females have these very specialized movable spines on the pedipalps that are set into this very deep socket. And the socket, as you see here, has an innervation that allows that movement of that spine, which was not thought to be apparent in any of the other genera. But using just SEM, she was able to demonstrate that we do see that same similar kind of innervation of the spine in Hemerotreca, in some species, maybe of Eremobatidae, but in Eremocheilus and Hemerotreca, whereas Eremocosta, Eremothera, Chambria, and Eremorax, the spines that we see are closely set and immovable. So this is an interesting avenue to explore and just demonstrates how much information there is in these morphological structures that we have yet to explore. Ryan has just completed a project looking at the phylogenetic significance of these ctenidia on the underside of the abdomen of males, because the ctenidia have been used, these specialized spine-like structures, or specialized setae is what they are, have been used as diagnostic characters, particularly the number of ctenidia, the presence or absence of ctenidia, the number of ctenidia, the shape of ctenidia, and the length of ctenidia. So Ryan analyzed over 800 specimens representing as many species that had ctenidia as he could find in the collection, and representing multiple specimens per species to get an idea of the diversity and how variable these characters were intraspecifically. And what he found is that ctenidia number has a phylogenetic signal, but shape and length do not. So he's just finished this project. This was the bulk of his master's work, and this is ready for submission. We are also hoping to explore characters associated with the chelicerae, because the chelicerae in Solifugae, if you looked at the posters that are presented by other labs or some of the talks that were presented in this Congress, you know that the chelicerae are sexually dimorphic, and particularly in the family Eremobatidae. Here we see male and female chelicerae, male and female chelicerae. And what you should notice is that on the fixed finger of the male chelicerae in Eremobatidae, there are no teeth, whereas females have teeth on the upper and lower, the fixed finger and the movable finger, there are teeth. for the females, no teeth for the males. And oftentimes you see other modifications of the male chelicerae, as you see in this species of Chambria. Or you, even in all of the other families, we also see modifications in the male chelicerae. So these are the chelicerae in the family of Amortrechidee, and you can see a specialized structure here called the flagellum. And in some of the old world families, other modifications of the male chelicerae and a weird looking flagellum here. We have no idea what those flagella are functioning as, but the jaws, the chelicerae, are used in mating and copulation. So what the males do across the board for all the different families is they use the jaws, they use the chelicerae to place sperm into the female's gonopore. And that's what we see in
Paula,
this view. This is a male.
5 minutes.
Okay. This is a male that has his chelicerae deeply inserted into the female's gonopore. And I will show you this video. This is a video that another student I worked with, Jenny Rousel, produced. And in this video, you will see Eremobatidae copulation. And what you're going to, this is the female running around, and you're going to see the male rushing at her. Here comes the male, very anxious. He grabs her. What's interesting in the Eremobatids is that as soon as he makes contact, the female goes into a state of lethargy. She completely stops moving. We have no idea what triggers that cessation of behavior. Then the male, and Jenny was able to, Jenny was able to demonstrate that the male after he puts her into this trance-like state, the male will then use his suctoral organs to maneuver, and he inserts his fixed finger of his chelicerae deep into her gonopore and begins a chewing motion. And he continues that chewing motion for a good 1.5 minutes or 2 minutes. And then what you're going to see with the male is that he will very suddenly release, pull his chelicerae out of her gonopore, move his body up, and directly deposit a sperm packet directly onto her gonopore from his own genital opening. And then he moves his body back down and pushes that sperm packet right into her. We're not sure what's going on here. We think that in the initial chewing, maybe he's either preparing her for receiving his sperm and or removing sperm from competing males. And then in that second chewing that we see, when he pushes the packet in, clearly he's using his chelicerae to push the sperm packet in. And then the continual chewing may be opening and releasing the sperm packet. But he's about to move up and deposit he deposits his sperm packet, and then he puts his chelicerae back into her and chews on her again and pushes that sperm packet back into her. Now, if you watch the female, she will begin to wake up. And as soon as she begins to wake up, her chelicerae start to move. She starts to move her legs. And at that point, very soon after that, the male pulls out and runs away because cannibalism is very high. I'm going to skip, we're doing some work. Erica and Ryan did some work on a mark recapture study that demonstrated that there seems to be site fidelity, which I can talk about more if you're interested. And we're also enhancing our online presence by using our undergraduate students to create an online field guide to Eremobatids by linking very high quality photographs with the records. And this is using the Scan Symbiota database. And then Warren, of course, is updating our Solifugae website. So I'll end with that, and hopefully we have a few minutes for questions.
Wow, that was really interesting, Paula. Thank you. That's a group that I don't know much about, so I've learned a lot in that time. Prashant has a question, so I will ask Prashant to unmute and he can ask.
Paula,
You should
thank
be
you
able
for
to.
an awesome